Hey, so this is my lecture on really the cell membrane and passive cellular transport. So we're going to define cellular transport, describe the structure of the cell membrane and how molecules move through it via simple diffusion, define surface area to volume ratio. That's really why cells are small. And then you're going to calculate the surface area to volume ratio for a hypothetical cell and explain why surface area to volume ratio is important to the survival of the cell, define osmosis, describe how environments affect the water content of the cell, and describe facilitated diffusion. These are the key vocabulary words. So as cells perform their function, they need to absorb nutrients and food from their environment. So they need to take some things in and they need to take things out. So the cell membrane is sort of the gate, you could say. It lets things in and it lets things out. Okay, so that's what I just said. Okay, so this is the cell membrane. So we're going to talk about the cell membrane first, and it is that lipid bilayer, and it's a phospholipid bilayer. And we already did the phospholipid when we did the macromolecules. So one of the macromolecules were lipids, and one of the example we gave was the phospholipid. So this is a phospholipid. It has a phosphate head. It usually has a charge on it, which makes it hydrophilic, which means it likes water. And these are fatty acid. These are those long carbon hydrogen chains, and if they're bent, um, they're not they're unsaturated. But anyways, so these are nonpolar because there's really no oxygen or no charge associated with these. So that makes them hydrophobic, which means they don't like water. So this likes water. This doesn't like water. It's amphipathic is the word for that. But anyway, so that's that. So this is the hydrophilic head, which is a phosphate head. And then this is the lipid tails, which are hydrophobic. And they are going to line up this way, right? And that's why it's a bilayer, is because there's two layers, right? There's two layers, and they're going to line up so that the lipids are in the inside because they don't like water, because our cells are basically bathed in water. And the phosphate heads are on the outside. And basically, all our membranes look like this. So the membrane in the mitochondria, the membrane in the endoplasmic reticulum, the nuclear membrane, the vesicle membranes, all the membranes in our cells are basically phospholipids. So plants also have this phospholipid, but on the outside, they also have a plant cell wall. But this is a cell membrane or a plasma membrane. You can call it either, whichever one you want. Okay. So that this is the way they're lined up, and it's a bilayer because there's two of them. And so basically, since this has a charge, right, this is going to like water. This doesn't like water, and this likes water. So water basically can't pass through here because it's polar, and it can't pass through this nonpolar region. So the only thing that can pass through here are either small or nonpolar molecules. And actually, water can't pass through there. This is wrong. Water can't pass through there because water is charged. So, but oxygen can, CO2 can, hydrogen ions can, even though they're charged, they're small. And so it's selectively permeable. So what it lets through is small nonpolar molecules. Everything else is going to need some help to get through. So this is outside the cell. This is inside the cell. Right, so these have to be not small nonpolar molecules to move through there. Okay, so we'll skip that. Okay, so we'll skip that part. Okay, so if you are not, if you're trying to get something bigger or polar through there, you actually are going to have another way to do that. And so like glucose is a bigger molecule, salt is a bigger molecule, fats are a bigger molecule. All these other ones are actually bigger molecules, and they're going to need ways to get through there. So these are the parts of the other parts of the cell membrane, And they call this the fluid mosaic model because it actually sort of moves around. It's very fluid, and these proteins can move around in it. And I might show a video of that. I'm not sure. Okay. So I'm just going to go through the parts of this, and we would have made a model like this in class. So anyways, but here is cholesterol. And basically most of your cholesterol is made by your liver. And, most people, if you go and you have high cholesterol in your blood, they want to put you on medicine. But cholesterol is actually an important part of your cell membrane. So it actually is a fat, so it fits right in here. So it will actually embed into this membrane because this is nonpolar and this is nonpolar. They're both nonpolar, both fats. And so this is really important to keep in the fluidity of your membrane. So it has to keep moving because it's a fluid mosaic. So fluid means it's moving, and mosaic means it has different things in it. But cholesterol, like if it gets real cold, the cholesterol prevents your cell from freezing up. If it gets real hot, it sort of holds your membrane together. So this is really important for sort of the structure of the membrane and maintaining the structure of the membrane. Okay. And different types of cells have more cholesterol. Other types of cells have less cholesterol. So it varies depending on what type of cell you are. Okay, so glycolipids. So this is a glycolipid. So glyco is a sugar. So glyco means sugar. So, or a carbohydrate. You could also say it's so carbohydrate is also a sugar. So you have glycolipids and you have glycoproteins, and this is called a carbohydrate. So this is a carbohydrate on here. Here's a carbohydrate on here. Here's a carbohydrate here. Here's a carbohydrate, and here's a carbohydrate. So they're all carbohydrates or sugars. And that's where you get that glycoprotein and glycolipid. So glycolipid is the carbohydrate is attached right to the lipid. Instead of the phosphate head is sort of gone and it's attached to that lipid. So this is important for recognizing other cells. So this is cell recognition. So if it doesn't recognize you, it will try to fight you. So this is important for like if you get a liver transplant or something. So they make sure your glycolipids match up so you don't reject that the donated organ. That's the idea. All right. So glycoproteins are essential for cell-cell communication and assist with the transport of molecules through the cell membrane. So this is how they communicate. But again, they do this. This is a protein and it has a carbohydrate on it. And generally, these are a certain they're a certain branch or something. So they're each one is different, and they communicate differently depending on how they're built and how they're branched and things like that. So there's all kinds of different glycoproteins. There's all kinds of different glycolipids. So this is just a class of proteins. This is just a class of these, but there's all kinds of different ones that are in your cells, and depending on what cell you have determines on how many and what kind of glycoproteins and glycolipids you have. Okay, a peripheral protein maintains the cell's cytoskeleton. So the cytoskeleton sort of keeps its shape. So it keeps its shape. And that is this right here. So peripheral proteins don't go all the way through the membrane. They're just here on the side. So that's a peripheral protein. Okay, so these are channel or integral proteins. So they do span the length of the membrane. So they generally stick out the top and they stick out the bottom. And the parts that stick out the top and the bottom are generally polar. The parts that are in here, they fold, and this would be the nonpolar parts, right? So proteins have to fold. So they're made of amino acids, but they have to fold. And when they fold, they'll fold so that the nonpolar parts will be on the outside. And actually, if you're going to let water or something come through here, the inside of it will have to be polar. And then these sides that stick out the other end will also be polar because it's bathed in water, and water is polar. And the same thing here. So this is sort of the same thing. This is a channel protein. This is sort of like a gate, so it sort of opens and closes. These can also open and close, but they basically let things through. This is a globular protein, which also acts as transporters through. Okay, I'll skip that one. That one's not quite so important. And so structural proteins, that's like kind of like the cytoskeleton and stuff like that. That hooks to that. That's actually not shown on here. These alpha helix proteins, so this is a protein that is folded, and they basically plug gaps and stuff in the phospholipid bilayer. So the bilayer is very flexible, but it does get gaps, and then depending on what kind of cell it is also. Okay, so if you want to know the functions, so cholesterol here would prevents stiffness. The alpha helix proteins would plug gaps. The glycoproteins are for cell-cell communication. The glycolipids are for cell-cell recognition, and the channel proteins are for transporting molecules. If you look here, that should be right. Okay. So that's sort of the parts of the cell membrane, but then again, that's called the fluid mosaic model. So this is called passive transport, and the biggest thing about passive transport is it doesn't require energy. So it's passive, so it requires no energy. And basically it works on a concentration gradient. So basically your cell has to have concentration gradients to move things across the gradient. And the way I would compare this is to like a battery. So when you buy a new battery, all the electrons are on one end, and we'll say all the protons are on the other end. So you have a gradient. And then when you hook that up to your earphones, it allows the electrons to flow over to the other side, and they will flow over to the other side until they are at equilibrium. And then there's no more electrons will move over. So your battery isn't specifically dead, but it lost its concentration gradient. So it's at equilibrium, which means it's not going to produce any more energy. So that's the idea. That's how I would think about a concentration gradient. Okay, so basically this has a high concentration. Let's call them hydrogen ions because that's what we're going to use eventually. And then this would be a low concentration of hydrogen ions. Okay, and this would be a big concentration gradient because you have a high concentration here and a low concentration there. Okay, so there are three types of passive transport which are used to move particles in and out of the cell. So I'm going to tell you right now, when you talk about osmosis, we're going to talk about water. This is water. This is water moving. But we're going to do diffusion first. All right, so in which direction do molecules usually move? They move from high concentration to low concentration. What does passive cellular transport mean? It means it relies on a gradient because it doesn't use energy. So it relies on that gradient to actually drive it. And then the three types are diffusion, osmosis, and facilitated diffusion. Okay, so diffusion is the net movement of small or uncharged particles from an area of high concentration to an area of low concentration. And this small uncharged means that's what's going to diffuse across the cell membrane because it has to go through that lipid layer, which is nonpolar. So they're uncharged. And this is just showing you here. So if you have a beaker of water and you put in a sugar cube, so again, a sugar has all those oxygens on it, which makes it polar. And water is polar. So like dissolves like. So sugar does in fact dissolve in water. And eventually it's going to start dissolving and it's going to dissolve more until actually it comes to equilibrium. So this is at equilibrium where there's equal amounts of sugar and equal amounts of water and they're equally distributed. So that's equilibrium there. And so here, right here, you have a different it's in equilibrium. Here you have equilibrium, so it's not going to move anymore here. Okay, so this is where you have to have small nonpolar will pass through here. If it's large or polar, it will not move through here. And so here is outside, and eventually, if this is a nonpolar molecule, it will start moving across the lipid bilayer, or the cell membrane, or the plasma membrane, until you reach equilibrium. So this is over time. All right. Okay, so that's what this is saying. So they're just going to keep moving until they get to equilibrium. So this should be not small nonpolar molecules are going to move across, all right, and large polar molecules are not going to move across. A higher temperature makes molecules move faster, and if it's a very thick membrane, it means it's going to move slower. And cell surface area. So the greater the surface area of the cell means more area available for diffusion. So that's increasing your surface area. And I'll skip over this. But equilibrium is basically they have the same concentration throughout the whole thing. Okay. So I'm going to talk a little bit about surface area to volume ratio, and you could put this other places. Some people put this where Y cells are small, and so you can it sort of can go both places. But so the surface area of a cell is the area that is in contact with the extracellular space, and generally you think of cells being kind of round or square. But if you think about a neuron is really long and skinny. And as the cell increases its size, its surface area will increase accordingly, allowing for greater diffusion. However, this change in size also changes the cell volume. In fact, increasing the size of the cell has a much greater effect on the volume of the cell than it does its surface area. So just making the cell bigger increases the surface area, but it doesn't increase the surface to volume ratio. So anyways, I'll skip over this. So, okay, the surface area to volume ratio of a cell is defined as the area of the cell's surface compared to the area inside the cell. So smaller cells have a much greater surface area to volume ratio, which allows for the efficient diffusion of substances through the entire volume of the cell. So, and gracefully to increase surface area, you're going to fold it, is what you're going to do. So a good example here is this. So we talked about the endosymbiosis, and this is the mitochondria. So it has a double membrane, right? So this is the membrane that prokaryote had, and then this is the membrane, and these are phospholipid bilayers, and then this is the membrane it got when it entered the cell through endosymbiosis. So it has a double membrane, but you can see that this membrane has all these folds, right? And all these folds give it a lot more surface area. So it's increasing surface, and this is important for this particularly because it makes ATP. So it actually makes ATP across this membrane right here. And you have to rely on concentration gradients and things like that. But the idea is that you have more surface area because of the folding on that membrane will allow you then to make more ATP. That's the idea. Okay, I think I said everything there. And this is the pili of an intestinal cell, right? And you can see that all of these things actually increase the surface area, right? So if you're eating food or whatever like that, you have all this surface area to actually take in the food and, things like that. So surface area cells have all kinds of ways to increase their surface area. And this is how you calculate that. We would have done a worksheet with this in class. So I'm not sure how much I'm going to do here, but So basically the surface area is the 6 times the area of a single size if you're talking about a cube. And the volume is width times depth times height. And then the surface area to volume ratio is just this number divided by this number, right? So it's surface area divided by volume is what it is. So that would be 6 to 1. Or if you put 6 over 1, it would be 6. So this is just going to show you that here is the surface area. So what cells are trying to do is maximize this right here. So they're maximizing the surface area to volume ratio. And you can see right here that the smaller the cell is, the higher the surface area to volume ratio it is. If you get bigger, you actually have more volume and you're lowering your surface area to volume ratio. So this is important for letting things in and out of the cell. So the more surface you have, the more, peripheral proteins and channel proteins and integral proteins and glycoproteins and all that stuff you can have. And so the surface area here would be 6 sides, right? And 6 times 1 is 6. And the volume would be length times width times height. That would be 1 times 1 times 1 is 1. And then this is the surface area here. This would be 2 times, I can't remember how this was. How did this go now? This is 6. I cannot remember this now. I don't know if these numbers are actually right. They don't look right to me. Anyways, okay, I'll just keep on. So how does surface area affect diffusion? So the greater the surface area allows more molecules to diffuse into and out of the cell at the same time, increasing efficiency. That's what you really need to know. That's the end result. I actually don't think I'm going to have you calculate surface area to volume ratio, but you should know that the higher the surface area to volume ratio, the better. So that's better for the cell because it has more surface area to move things in and out of the cell. Okay, so I'll skip over that. All right, so I'm going to talk about osmosis and facilitated diffusion. So osmosis is the diffusion of water through the partially permeable membrane from a region of high concentration to a region of low concentration. So water, when you talk about osmosis, you're talking about water, right? This is important that you're talking about water. And I'm actually going to give you a little lab on that. And I'll talk about the lab later. But we would have done the potato lab here. Okay, so, but this is a selectively permeable membrane right here. And you could think of this as a cell membrane, but it's not a cell membrane. But anyways, you have sugar water over here, and you have less concentrated sugar water over here. So there's more water here and less water here, right? So you're talking about water. When you're talking about osmosis, you're talking about water. So you're not looking at the solutes, you're looking at the water. So since there is more water here and less water here, water should move that way. So it's going to move, the water should move that way. Okay. All right. All right. So, So cell membranes actually are not permeable to water. They actually have to have a protein, an algoporin, to move across the membrane, but water can move across the membrane. That's actually not true there. So so there's 3 types of solution that involve water, each which affects the cell differently. And basically there's hypertonic, isotonic, and hypotonic. So hypertonic solution, so it's really talking about the solution. And generally when we would have done water potential problems here. And the way you set up water potential problems is you put cell in a beaker of water or sugar water or something, right? And you can think about this is the beaker, and you're kind of looking down on the beaker. So here it says there's a higher concentration of sugar in the solution, right here, this is the solution, and then these are the cells. So if there's more sugar in the solution than in the cells, then water is going to move where there's less water. So there's more sugar, less water, less sugar, more water, and water is going to move out into the solution, which makes it hypertonic, right? So water flows from an area of high concentration, so there's more water in the cells, to an area of low concentration, which is outside the cells, and then the cells are going to lose water and lose weight, and they're going to shrivel. And animal cells, so this is a plant cell wall. And plant cell walls, I think plant cells have those big vacuoles. And those vacuoles have to be full of water in order for them to push. It's called turgor pressure or pressure, and it pushes up against that cell wall. This is a plant cell right here. So if it has plenty of water, that vacuole is going to be full, and it's going to push up against that cell wall, and that's what this is showing you here. All right, so that's when water is flowing in. But if you put a plant in, say, salt water or something like that, or sugar water, basically there's less sugar. There'd be less sugar outside, more sugar outside, and less sugar inside, and then the water would move out. And that's when this is plasmolysis, when that plasma membrane moves off of the cell wall, and this is when the plant will kind of get limp and kind of fold over and things like that. What do you call that word? I can't think of the word. Anyways, okay. So a hypotonic solution. So I will tell you, water is the most hypotonic solution. And the solution contains a higher concentration of water than the cell. So this is the solution, right? And you could think of the hypo as this O. So you could always think of water. Hypo is pure water. So pure water is here. And so there's more water outside than inside. So the water should move into the cell. So whenever it says pure water, you know that is a hypotonic solution, and water is always going to move into the cells because water is as hypotonic as you can get. So basically it's going to keep flowing inside the cell until eventually it busts for us. For plant cell walls, they actually have, for plants, they have cell walls, which prevents them from doing that. In animal cells, the cells burst or will lyse, killing the cell. This is because they lack the support. Okay, so that's what I just said. That's what I just said there. So this has a cell wall. Okay, an isotonic solution is really where you want to be. So you don't want your cells to take on water, and you don't want them to lose water. Right? You want them to be isotonic. And basically, if under normal conditions, they are, because your cells are healthy. And that means that there's an equal amount of water outside and inside the cells. So water can still move through by those aquaporins, which is that protein, but there's no net change in weight. Okay? So here's no net change in weight. Here, the cell is going to gain weight, and here the cell is going to lose weight. And that's going to be important when you do that lab to understand that right there. So if it's in a hypertonic solution, it's going to lose weight. The cell will lose weight. If it's an isotonic solution, the cell will not gain or lose weight, or the weight will stay the same. And if it's a hypotonic solution, the cell will gain weight. And that's what that lab is going to give you, is weights. Okay. Okay, so that's what this is saying is in animal plant cells, this keeps its shape when it's in an isotonic solution. So this is what your cells prefer and where they actually survive the best. All right. Okay, so this is a comprehension test. So basically, this is cell A. So this is 0% sugar in the solution, which means it's in pure water. So this is hypotonic. So water, in a hypotonic solution, water is going to move into the cell. So right here, the water should move into the cell, it should gain weight, and it should basically look like that. Okay, so this one has 10% sugar. This has the least amount of water. And so water here should move out. So water should move out, and this actually should get smaller. And so it should look actually like this one. It should look like this one. Oh no, it should look like this one. I'm sorry. This is 10%, right? So this one should look like this one. And then this one should be in the center. This is basically considered isotonic, I guess. They're saying this is isotonic, and this one is going to look the same. Okay, so here water is going to move in. Here water is going to move out. Here water will move in and out, but there's going to be no net change. So this is hypotonic. This is hypertonic. This is isotonic. I think that's what that says there, hopefully. Okay, so facilitated diffusion. So some molecules, such as sucrose, are too large to pass through the cell membrane by simple diffusion. These molecules require assistance of other molecules, such as channel or carrier proteins, which are in the cell membrane, right? So that's this integral protein or a channel protein. And they can work kind of like gates, right? So this I would call a gate, where the sugar molecule, whatever the cell needs, is going to come in here, and then it's going to close, and it's going to have a swivel, right? And then it's going to swivel back up this way. It's going to take in another one, right? And so you're still moving it along the concentration gradient, but it's too big or it's polar and it can't move through the plasma membrane. So it has to have a channel protein. But again, this is done with no energy. Okay, so I think I just said that. Okay, we'll do this true or false. So facilitated diffusion expends energy. That is false. Facilitated diffusion uses glycoproteins to transport molecules through the cell membrane. That is true. This is true. It moves it down the concentration gradient. That's also true. And this is also true. Yeah, so that should be true. This one was false. I don't know why that one's false. Okay. So I think that's it. So that's all there is for that right there. And so I will put the next one on in a little while.